Okay, hello, and welcome to day 2 of our week-long lunchtime series, Ethical Issues in Pediatric Gene Therapy Clinical Trials, happening each day from noon to 1 p.m. through this Friday, December 4th. Today's installment is entitled Equity Issues in Pediatric Gene Therapy Research. We invite you to join us tomorrow for Ethical Challenges of Immunogenicity and Toxicity in Pediatric Gene Therapy Research. If you haven't registered already, registration is still open for the rest of the week. More information can be found at our Eventbrite page, and we'll drop the URL in the chat. This lecture series is brought to you by the NYU Grossman School of Medicine, Division of Medical Ethics Working Group on Pediatric Gene Therapy and Medical Ethics, also known as PGTME. You can find more information about PGTME on its website, and we'll also drop the URL in our chat box, or you can just Google NYU PGTME. You can also sign up for PGTME's quarterly newsletter to stay abreast of our work. We will also let you know when this webinar, along with the other webinars this week, will be uploaded to our YouTube channel. And yes, that means that this event is being recorded. We regret that we are unable to offer real-time transcription of this event, but the archived videos will have captions. My name is Aisha Langford, and I'm an assistant professor in the NYU Grossman School of Medicine, Department of Population Health, and a faculty member of PGTME. Today's event will first feature a presentation by Dr. Pilar Osorio, who I will formally introduce momentarily. During Dr. Osorio's presentation, please submit your questions via the Q&A feature at the bottom of the screen. After the presentation, there will be a brief response by Dr. Rafael Escandon, who I will also introduce momentarily. After that, I will pose the questions you have submitted via the Q&A feature to both of our speakers until we run out of time. And without further ado, I would like to introduce our first respondent and then our speaker. Dr. Rafael Escandon is the Senior Vice President of Medical Affairs, Policy, and Patient Engagement at BridgeBio in San Francisco, California. Dr. Escandon has worked in global clinical research and development and patient advocacy in the biotechnology industry for the past 25 years. He has also served as an adjunct faculty member at the University of California, Berkeley's Clinical Research Management Program, and engages frequently as an educator on causes related to human rights and justice. Our initial speaker is Dr. Pilar Osorio, Professor of Law and Bioethics at the University of Wisconsin-Madison, and co-director of the Law and Neuroscience Program. Formerly, she was the director at the Institute for Ethics at the American Medical Association, and worked as staff for the federal government's program on ethical, legal, and social implications of human genomics. Dr. Osorio has served on numerous federal and international science policy advisory committees. Her research interests include governance of large-scale biosciences research, protections for people who participate in research, data sharing, ethics of big data science, and uses of race in research and medicine. And on that note, I would like to thank Dr. Osorio for joining us today, and I will turn the mic over to her.
Thank you and welcome. Let me just start sharing my slides here. Uh-oh. Are they sharing?
Yes.
There we go. All right. So hello, everybody, and welcome also. First, I want to thank Aisha and other organizers of this set of events for inviting me to speak. I'm thrilled, and I attended Dr. Mark's talk yesterday, and I look forward to attending some of the other talks later in the week. So today we're going to discuss equity in gene therapy, and I'm sorry to say that my examples and my focus is not so much on pediatric gene therapy, although it's not specific necessarily to pediatric gene therapy. And I do have one example that is specific to pediatric gene therapy, but I might not have time to go into that example. So I do apologize if this seems like a bit of false advertising here. So before I get started, I wanted to make a disclosure, which is that in the past year, I've done bioethics consulting for 2 pharmaceutical companies, Eli Lilly and Genentech. Actually, it's all sort of general bioethics policy, nothing specific to gene therapy or cell therapy, but you should take my comments with this in mind. So as we start out, of course, since we use the word equity in the title, we ought to think about what that means. I actually prefer to focus on situations where we think there might be a lack of equity in health, healthcare, or biomedical research. I think it's often easier to agree on situations where there is inequity that ought to be ameliorated rather than on what counts as equity. So when I talk about inequity, I'm really thinking about a situation or state of affairs where there is unfair inequality. And there are, of course, many theories and definitions of fairness, and I'm not going to go into them here, but suffice it to say that in many cases, people can have different theories of fairness and still agree that a particular health disparity or a particular arrangement in biomedical research constitutes an unfair inequality. And so if we're talking about unfair inequalities in health, here is an oldie but goodie definition, right? So, and what I like about this definition, of course, it does incorporate my notion of unfair inequality, but it also focuses on comparison between groups of people, which is what most of us who think about equity are concerned about. And it focuses on the fact that some groups are systematically disadvantaged with respect to opportunities to be healthy. So what do we mean by systematically disadvantaged? We mean, I think, that the operation of relevant institutions, including but not limited to research institutions and healthcare institutions, the operation of institutions consistently undermines some people's opportunities to be healthy as other people or to achieve a sufficient level of health. I also think when people use the term equity, they're thinking about trying to achieve certain types of equality, which is also reflected in this definition. People are thinking about equality of opportunity for something. And in this case, this definition focuses on equality of opportunity to be healthy. In research, we might also think about equality of opportunity to be studied or to have your problem studied. So overall, I think this is a reasonable way to think about the concerns that we focus on when we're thinking about inequity. So this definition talks about comparing social groups or groups with particular social characteristics. There are a lot of ways to group people. So which groups matter? I would say the groups that matter are the ones that matter most are the ones that are relevant for creating or constituting hierarchies of privilege and power in our society. So some groups of people have more privilege and power in the hierarchy, and some groups have less. And so the ones that have less are marginalized and oppressed in many cases. And so when we think about groups that are marginalized and oppressed in society, those are the groups that have systematically less opportunity for a reasonable level of health. And I think the groups that matter here are groups like racial groups, groups based on socioeconomic class, gender, actually urban and rural groupings of people matter for health and healthcare. There are other ones. My list is not an exhaustive list. I think the point here is that the way society functions systematically advantages some people with respect to health and disadvantages other people with respect to health. And we should focus on the fact that some groups of people are systematically disadvantaged with respect to health. And why does that matter? Well, because the living conditions and lived experiences of people who are systematically disadvantaged include actually many factors that come together to undermine their health. And these can be low education, low income, more dangerous working environments, more exposures to toxins in their home environments, experiences of stressors such as racism and other stressors that undermine people's health. So there are a variety of different dimensions of the social and lived experience that contribute to our health. And along many of those dimensions, people who are marginalized or part of oppressed groups will fare worse. Also, people who are marginalized or oppressed tend to fare worse in the healthcare system. That is, the healthcare system is less attentive to their problems, may treat them with racism or other forms of prejudice. And so we know that in every society, people in marginalized groups on average have less good health than people in more privileged groups. And that's not simply an accident or a fact of nature, it's something that we cause, right? Our social arrangements and institutions cause that to happen. We should also focus on people in marginalized groups because unfair inequalities in health and healthcare and access to health-related research are more likely to combine with other forms of disadvantage for such people and to exacerbate overall social inequality and injustice. So to the extent that we think about opportunity for a reasonable level of health as being important to achieving other goals and taking advantage of other opportunities in society, this is one, focusing on health is also helping us to focus on larger social justice questions. And so if we focus on, research that might improve the health for disadvantaged people or people who are medically underserved and more marginalized, then we not only can improve health, but we can improve social justice overall. So our response to unfair inequalities should be to remediate or compensate for them, where remediation involves improving the health of the group that is experiencing the unfairly poor health, so that the inequality lessens or disappears. If we can make that inequality disappear altogether, then that's complete remediation. That might not be possible in many health situations. They might be only partially remediable, but partial remediation is still a goal that we should really fight for that. That would still improve potentially social justice overall. Compensating means providing people with other kinds of advantages to offset health disadvantage. Many health inequalities are not really capable of being compensated. So other forms of unjust inequality might be more amenable to compensation. But in particular, if a person is, if there's some kind of unjust inequality associated with the fact that a child has a deadly disease and is likely to die in infancy or early childhood, there's no way to compensate for that. Instead, we have to focus on trying to remediate in a way that might extend life or help to ameliorate the impacts of the disease, right? And research is important here because it's actually how we move health conditions from a situation where there is no way to remediate them to a situation where we have some forms of remediation, right? That's sort of a fundamental goal of biomedical research. And so on the other hand, if we do our biomedical research in ways where the research itself is unjust and may have unfair inequalities built into it, then we can exacerbate health inequalities and we can exacerbate social injustice overall. So I just want to point out that the way we go about ameliorating unfair inequalities matters. And for instance, one could try to create a more equal situation by actually decreasing the health of a more privileged or healthier group, that would, it's called leveling down. That would not be okay, right? So we have to take account of other important ethical values and norms. Fairness and justice are not the only ethically important thing here. And as we go about trying to ameliorate health inequalities, unfair health inequalities, we have to think about how to do the amelioration in also an ethical manner. So before I move on to talking specifically about gene therapy research, I just want to point out that there are a lot of conceptual challenges in thinking about unfair inequalities and injustice. These are topics that people have thought about for centuries. They're not easy. And so some of the kinds of questions that come up, which inequalities actually count as unfair, right? And people aren't always going to agree on the answer to that question. How do we measure, if we're talking about health inequalities, how do we measure health and compare it across groups? What metrics should we use, right? So there are many, many of these kinds of questions. And, I just want to point out that it behooves us to recognize that reasonable and thoughtful people can come to different and well-justified conclusions in answering these questions. So I don't want to make this seem like it's a simple area. We are not going to spend time delving into any of these questions now. We're going to move on to talking about gene therapy research and some equity issues in gene therapy research. So some of the kinds of questions that we need to be asking really in any kind of research that we're doing, who do we study? And so that goes all the way to the preclinical and discovery phase. Whose cells do we use? Whose DNA do we analyze? And it is well known in genetics. I'm not going to go into it here, but there are a number of very good papers demonstrating that the vast majority of people who we've studied in our human genome research have ancestry from Northern Europe. Those people, it's wonderful that they're being studied, but the genetic diversity that they represent is only a fraction of the genetic diversity in the human race. And so that potentially could be a problem. But we also need to think not only about who do we study in preclinical science and then in our clinical trials, but why does it matter who we include? Because I just want to point out that inclusion of marginalized groups in research is not necessarily an unmitigated good. We can include people in ways that, for instance, reinforce racial stereotypes or stigma and ultimately add to people's oppression or marginalization. And this is particularly true with anything related to genetics because many pernicious racial ideologies have to do with notions of genetic superiority or inferiority. And so, I know genome scientists who were shocked to discover that their research is actually featured on Nazi websites and white supremacist websites. So, we have to keep in mind that researchers are not experts in things like race and racial hierarchy. They are not experts in, genome researchers, for instance, and people who are trying to develop gene therapies are not experts in, how to measure and think about socioeconomic class and or socioeconomic status and its relation to people's health. And so, part of this is why does it matter who we include? That's why does it matter who we include in research, but also why does it matter who we include as researchers? And perhaps that we need to include diverse groups of people with diverse expertise to help make sure that if we're going to include diverse study populations, we do it in ways that are helpful ultimately and not overall harmful. We have other questions like whose problems do we prioritize? How do we design our studies in a way that makes it possible and desirable for diverse study populations to participate? Again, there are lots and lots of questions here. These questions are not unique to gene therapy research, but they're really important. A question that is more focused on gene therapy research, how does the manufacturing process for gene therapy influence fairness among trial participants? Who does the actually, I should say, how does the manufacturing process influence fair access when the gene therapy product comes to the market? So because manufacturing is very complicated technically, there's a lot of regulation around it. Not everybody's, gene therapy is not going to work for everybody and in every case. So how do we optimize so that we can get, sort of the most people helped by this? I think those are questions that are very understudied. And perhaps Raphael has some ideas about those questions. All right, so I don't have time to discuss all of the relevant issues here, not by a long shot. So I'm going to give you two examples of sort of equity issues relating to gene therapy and more broadly relating to research. So one set of issues has to do with the question of whose problems do we prioritize? And I want to use the example of investment in sickle cell research as compared to investment in cystic fibrosis research. And I'm using these two conditions, both because people have actually studied this and analyzed in some detail. So we have data on investment in research for these two conditions, but also because in the last 3 to 4 years, the NIH and some companies have begun clinical trials of gene therapies for sickle cell. And I believe there are also clinical trials of gene therapies for CF. So both CF and sickle cell are caused by mutations in a single gene, and their inheritance is recessive. Both diseases cause significant morbidity. Sickle cell can be extraordinarily painful, and both diseases are associated with substantial reduction in a person's median lifespan if they have the disease. I would note that in the US, the lifespan for people with cystic fibrosis has gone up substantially since the 1990s, whereas the lifespan for people with sickle cell has either stayed the same or, according to some reports, decreased by 4 to 6 years. Interestingly, sickle cell was the first disease described at the molecular level, which was done by Linus Pauling in 1949. So some 70 years after that, there are only 2 medications approved for treating symptoms of the disease, one of which was approved in 1998 and the other in 2017. For cystic fibrosis, I believe, so the gene that, there are many mutations of the CFTR gene that can cause cystic fibrosis. That gene was discovered much after the mutation that causes sickle cell was discovered. So there are at least 6 medications approved for treating CF now, 4 of which are precision medicines. So these aren't gene therapies, but they're small molecule drugs that target the protein malformations caused by particular mutations in the CFTR gene. And so these are therapies that deeply depend on our genomics research. So 2 medications for sickle cell, at least 6 for CF. In the US, sickle cell disease is often thought of as the disease of Black people. This is because many enslaved people were forcibly brought to this country from countries that were in the center, a sort of belt across the center of Africa, where the gene variant that causes sickle cell was and is prevalent. However, sickle cell gene variants are also at a high prevalence among people in the Arabian Peninsula, in India, and in Greece, among other places. And in fact, the prevalence in parts of India and Greece are as high as it is anywhere in Europe, in Africa. But because in the US, we have, proportionally more people of recent African descent who were brought here from countries where sickle cell alleles were prevalent, we in this country think of this as a disease of Black people often. Cystic fibrosis, on the other hand, is found in people of all ancestries, but it's much more prevalent in people of Northern European ancestry. And I've seen it described with language such as the most common life-limiting genetic disorder in populations of European ancestry or in Caucasian populations, basically in white people. So CF is associated with white people. Sickle cell disease is associated with Black people in this country. And as of this year, there were approximately 35,000 people in the US living with cystic fibrosis. For those people's treatment, there was a network of more than 100 comprehensive treatment centers. There were actually about 120, and a few more than 100 treat both adults and children. On the other hand, sickle cell disease is approximately 3 times more prevalent. So there are maybe about 100,000 people living with sickle cell. There is no comprehensive network of treatment centers for them, although there are some scattered independent treatment centers for them. So with that as a background, let us look at research investments. And I know that this is a complicated slide, and I don't know if you can see my pointer, but what this slide shows is it's looking at funding by NIH, so public funding in research on sickle cell and public funding in research on CF from the year 2008 to 2017. And then also foundation funding for research in sickle cell and research in cystic fibrosis. And the key points can be found here, if you can see my pointer, that there is per person with cystic fibrosis or CF, there's approximately 3 1/3 times more funding for cystic fibrosis research, federal funding for cystic fibrosis research than there is for sickle cell research. Beyond that, if we look at the foundation funding, the differential is substantially more extreme, with about 75 times more funding for cystic fibrosis research than sickle cell research. This study also found that there were substantially more scientific journal articles in peer-reviewed journals that were published with data about cystic fibrosis, and of course that there were more drugs approved for cystic fibrosis than for sickle cell disease. So their conclusion was that disparities in funding between sickle cell and cystic fibrosis studies were associated with less research productivity for sickle cell disease and less development of novel drugs. So we have a disease that actually affects more people, but gets less investment, has substantially fewer treatment options. And many of us would think of this as a kind of unfair inequality in the way we've prioritized, the way we've set our research priorities. And I want to emphasize that in many cases, it reflects completely understandable individual choices, for instance, with respect to foundation funding. People who have CF, maybe in their family, and who are more likely to have the privilege of being a white person, may have networks, social networks of people who are wealthier and can contribute to a foundation and so forth, right? This is not, it's just an operation of society and a function of social hierarchies as much as it is anybody's fault, right? But nonetheless, this is a kind of inequality in research that should not exist. So I have to say that over the last few years at the National Institutes of Health, there are people who work on blood disorders and who have lobbied very hard for more investment in the study of blood disorders, and particularly in sickle cell. And recently, as I said, in the last 4 to 5 years, there have been a number of gene therapy trials, and particularly cutting edge using gene editing, that the NIH has funded and that private companies have funded. And part of this was due to an explicit desire to invest in an area of research that where we had underinvestment, unfair underinvestment, people would say, many people would say. And so here's a report of a patient who was in a gene therapy trial for cystic fibrosis, or for sickle cell disease rather. here is a scientific report of the first patient who went through a gene therapy trial for sickle cell disease. And what I have to say is that, actually I'll go to the next slide, which just shows this is a company which is running gene therapy trials for both sickle cell disease and beta thalassemia, which is a closely related disease. So, you know, blood disorders are a kind of low-hanging fruit for gene and cell therapy because it's much easier to do studies where you take people's blood out of their body or some portion of it, you isolate the relevant cells, you genetically engineer or modify them in some way, you can do some quality checks on those cells, and then grow them up and put them back in people's bodies, right? It is much more practical to do that kind of work than it is to try and target a gene therapy to a site inside of somebody's body. So it's not surprising that NIH and private companies, as they are moving into gene therapy trials, would choose to focus on single gene disorders in the blood, right? That can be accessed in people's blood. Nonetheless, the sickle cell trials, as far as I can tell, they haven't published much data. They have appeared to have been slow to accrue participants, even though sickle cell is, for instance, not nearly as rare as something like Duchenne muscular dystrophy or, you know, Duchenne muscular dystrophy caused by a particular mutation, right? And yet they're trying to accrue patients, like 45 participants for a clinical trial, and they haven't been apparently successful in doing that. So number one, this I think is turning out to technically be more difficult to have gene therapy help people with sickle cell disease, perhaps, than people had imagined it would be, but also that there may be some problems with the trial designs. I have no inside knowledge about why these trials might be accruing slowly. Maybe there are high financial burdens to study participation, or maybe the study sites aren't easily accessible to people with sickle cell disease. But I guess, you know, my main worry is that even if we ultimately, and when we ultimately get a gene therapy that is successful and is licensed by the FDA, how on earth are patients really going to get it, right? Because gene therapy is complicated technically, it's expensive, it can't be done by everybody, it can't be delivered in your local community healthcare setting. And you're looking at a patient population who actually are you know, disproportionately on Medicaid, for example, not receiving their healthcare in sort of high-tech healthcare settings. And so I think we need some really creative thinking here. And, you know, part of this is a problem of our healthcare system. And in no way do I think that we should, you know, cease doing research in gene therapy for sickle cell because our healthcare system has a lot of problems actually delivering that. But we might ask what researchers and the FDA can do in, you know, conceptualizing studies and designing them, and in the regulatory review process, to help increase the likelihood that a gene therapy product for sickle cell would actually get to the people who need it. Because otherwise, we could be learning a lot as scientists about how to do gene therapy, but not actually creating, not actually ameliorating any social inequality or improving an unfair health inequality. And so actually, I had another example, but I'm just about out of time. And this was my example that focused on more on pediatric gene therapy trials. But so I'm just going to skip this example, and maybe we'll have time to come back to this in the Q&A. I do want to say that this example addressed the question of the cost of gene therapy trials. So the therapies themselves, when they come on the market, can be extremely expensive, as we heard yesterday, in the hundreds of thousands to millions of dollars. And so this is actually a report of a study on people attempting to crowdfund their access to a particular kind of gene therapy called CAR T cell therapy. In many cases, so during the time period studied here, during much of that time period, there was no approved therapy. So this crowdfunding was primarily, almost exclusively, people attempting to get funding to participate in clinical trials. And even after 2 therapies came on the market, still many of the attempts at crowdfunding were people trying to raise money to participate in clinical trials. Some of these trials require people to move to a different city, for instance, and stay there for up to 6 months. That's an enormous economic burden. And that kind of burden on people is going to preclude a lot of marginalized people from participating in this kind of research, right? So I am basically out of time. I will come to my conclusions, which is that, and they're not rosy ones exactly with respect to improving justice and using gene therapy to do it, right? So I think there's a low likelihood that gene therapy will do much to ameliorate unfair health inequalities in the near future. And you know, the complexity and costs of gene therapy clinical trials, even before we get to the therapies in the clinical trials, I think the complexity and costs make it difficult to include people who are suffering from unfair health inequalities, or people from marginalized populations that are underrepresented in research. So we do really need some creative thinking about how to fund these trials in a way that makes it more possible for, you know, marginalized and underrepresented people to be participants in these trials, and more creative thinking about trial design and so forth and so on. So with that, I'm going to stop and thank you very much and hope we have a lively conversation.
Thank you very much, Pilar. Really, really great presentation. And what I'm going to try to do is to pick up on the thread of creativity that you laid down in your presentation, because I think that's what's going to be very necessary as we go through the future of developing gene therapies for particularly rare conditions. So I'm going to share my screen here. Hopefully this will work. And unfortunately, I'm not in presentation mode because it tends to crash my Zoom. So if hopefully everyone can see my slides adequately here. So I'm Rafael Escandon. I'm the Senior VP of Medical Affairs, Policy and Patient Engagement at BridgeBio Gene Therapy. Our company is located in San Francisco, but many of us are remote these days, of course, and I actually am living now in the Seattle area. But what I want to talk about today a bit before I go into my disclosures here, hold on. I am a full-time employee of BridgeBio Pharma, of which BridgeBio Gene Therapy is a division. And I have been a member of the Pediatric Gene Therapy Medical Ethics Working Group since 2019, proudly. So I'm glad to be a part of this presentation today. And again, thanks to Dr. Osorio for her very, very informative and broad presentation. I'm going to zoom down a bit into, as briefly as I can, really into rare conditions and ultra rare conditions, because and gene therapy in those areas, because I think it's an area of research that's still very much in its infancy, and one that I think we can have hopes that will address some issues of some of the issues of inequalities and issues of justice that we you know have emerged and are emerging. Just to put a slightly positive light on this. These are some statements that I took from FDA's website with respect to Rare Disease Day, and just to set a little bit of the prevalence tone here. for those of us working in, particularly and relatively exclusively in rare conditions, there are about 7,000, maybe a few more rare conditions that affect a large proportion, about 10% of the patients in the United States have some sort of a rare condition. So collectively, they're not uncommon, but individually, they very much are. And the FDA has, for the past 25 years or 30 years, really put some incentives in place for companies to focus on rare conditions. And some companies have been quite successful in doing so over the years. Last year, for example, there were 22 novel drugs and biologics that were approved by FDA that were orphan. So that's great. That's showing a lot of activity and a lot of interest in patients with rare conditions. And it continues to, in terms of the number of products entering development, has consistently remained greater than 500 annually. So FDA's interest and the industry's interest in developing new therapies for rare conditions seems to have been established, maintained, and is growing, which is really good. I only put this slide up for one reason, and I think it's to inspire some real hope that things can start to go very quickly when the needs are really high. So this is a tweet, actually, which I think this is the first time I've ever put a tweet into a presentation before, from Eric Topol yesterday. And it really lays out the timeline from when SARS-CoV-2, the virus, was sequenced and made publicly available, when the first case was, now down to what's in the red rectangle, really kind of where are we in terms of a vaccine or a couple of potential vaccines that are new technologies, not gene therapy per se, but related. And it really shows, I think, a movement toward optimization and production and prioritization of a potential extremely high need new therapy in a really rapid on a rapid timeline. So I think I bring this up only because I think this provides some reason for hope, because 10 years ago, this never would have happened so quickly. 5 years ago, it may never have happened so quickly. And again, looking forward optimistically here, this hopefully will result in community immunity for COVID-19. So in that regard, I'm not really going to focus much on this slide, other than to say BridgeBio, the company that I work for, really takes a portfolio approach across rare conditions. BridgeBio Gene Therapy is down here, but what we try to do is to not be limited to focusing only on a larger commercial market within the rare disease space. Rather, if we are able to have a blend and essentially put a fair number of cards down, our probability of success in a, for example, a more prevalent rare disease may really allow us to drive development of programs in rarer or ultra rare conditions that would have remained neglected without a good financial engine. So what BridgeBio tries to do is to spread risk and to place essentially a lot of bets across many different rare conditions, Mendelian genetic rare conditions, and in the hopes that, number one, we can treat patients and develop new treatments that are innovative, but also optimize in the process. So zooming into way down now into some of the real issues that relate to equity, I think these are topics that we talk a lot about at PGTME. We talk about them every day at BridgeBio Gene Therapy. And this list is not limited to what you see here, but I think these are topics that I think may come up in our Q&A and ones that we want to try to address. But really, with gene therapies, some patients get locked out from participation, for example, just by prior exposure or seropositivity to an AAV vector, for example. So they don't know how they got exposed to a virus in the environment, but that's the virus that is engineered to carry the functional genetic payload. Your immune system would attack that. They've already been sensitized. So that's an inequity, if you will, from some patients who have a rare condition, precluding them from participation. The first in human dose escalation trials, Dr. Marks talked about this a bit yesterday, and it's a real conundrum. The way that those trials are arranged, and I'll go into that in just a moment, there is, for the first participants who receive the first dose, unless we're lucky, there's a possibility of no benefit. And so what we try to do is to really minimize the number of patients who receive a low or potentially ineffective dose, because they only get one shot at the gene therapy. Related to that, of course, is redosing and immunogenicity. Again, it's if the immune system has been exposed to a vector in the past, a similar one, the virus, then there's really no possibility that the immune system will attack that vector and render it unable to deliver its functional genetic payload. There are also age-related or development stage-related, depending on the rare indication, inclusion criteria. So there are windows of opportunity, if you will, which I'll go into on the next slide, that may and often do preclude patients who have a lower likelihood of benefit, not just in clinical trials, but also in an expanded access scenario. Committing to 5 to 15 years in a trial, Dr. Osorio just mentioned that you really have to be in a situation where you have the financial and the practical wherewithal to participate in any clinical trial. Well, gene therapy just raises that bar in terms of what you need to commit to. So it's a really important consideration, and one that really can preclude patients from participation. Also, a concern, of course, is that by getting a gene therapy, that can affect your subsequent participation, even in non-gene therapy trials, just because of the way that drug development works. If a patient has received a gene therapy that they've committed to that they've had to commit to a 5 to 15 year monitoring program, and that can preclude them from participating in other, even non-gene therapy trials, for both practical and sometimes impractical reasons. And then finally, supply constraints. There's a huge front-end investment in terms of being able to invest in gene therapy programs and to engineer a vector and to scale it up to make an adequate supply to not just to supply clinical trials, but also to supply patients that may request expanded access. So it's a real problem, and it's a real limitation. So on my next slide here, I just want to show you that this is a slide partly shown by Dr. Marks yesterday as well. And this is one of the 2 approved gene therapies in the United States. It's Zolgensma for spinal muscular atrophy. What you see on the left-hand side here is the low-dose cohort. The y-axis is actually the functional measure of efficacy. And what you can see is that the patients were all dosed here at a low dose, but they were all aged around 5 months or so. So what you can see, this is what I was talking about in the prior slide of patients who didn't receive any benefit. These lines really aren't going in the right direction here. So unfortunately, with this low dose, they didn't receive the benefit, and they're not able to get a higher dose. later because of the immunogenicity reasons I mentioned. On the right-hand side here, you can see what a real response looks like at the higher dose here in cohort 2. So again, I think this trial illustrates a lot of the challenges that we have, which is number one, there is a dose-related one-shot opportunity here that we have to really consider carefully. But the second is the age-related one. And Dr. Marks pointed this out yesterday, that this patient was a little bit older, received the higher dose. It's obviously quite compelling to see that how the effectiveness of this dose in every patient that received it who was less than 5 months old. And then here, unfortunately, you have this patient that was a little older and clearly just demonstrates a window of opportunity for a gene therapy. Finally, what I'll say is that, again, we're in our infancy here. And as Dr. Osorio pointed out, there are opportunities for us as sponsors of clinical trials, as regulators, as academics, as patient advocacy groups, to really move through and cooperate to maybe rev things up to the point that the mRNA vaccine groups were able to. I won't go into every single element here, but what we really have an opportunity to do is to advance the technologies, scale up and optimize, cooperate to find redosing strategies, share gene therapy-wide observations, come up with strategies to manage off-target effects, and share natural history data. And if we can do that as a community, I think we can really move things forward very quickly. So with that, I'll cede back to Aisha and the Q&A.
Great. Thank you so much, Rafael and Pilar. Those are great presentations. So we have a lot of great questions from the audience. So I'll start off with kind of broadly, there's been a lot of conversation in academic circles lately about when, if ever, it's appropriate to focus on race, ethnicity, since those are social constructs. And then kind of related, there was a question around kind of the geographic distribution of the over 300 pediatric gene therapy trials, and kind of how does that relate to some of these inequities? So kind of why do we study race, ethnicity when that's a social construct? And then how does geography kind of overlay with the equity issue? So we'll start with maybe Pilar. Yeah.
Okay. So I think I actually was trying to emphasize that social conditions and social hierarchy shape our health in all kinds of ways. So using those categories is not necessarily the wrong thing to do. Furthermore, living your life at a certain place in a social hierarchy can actually become it can be embodied, right? So it can change the genes that you express. The social conditions in which you live can affect the genes that you express. It can actually poison you, right? In my opinion, we shouldn't use race or ethnicity as a proxy for other things that are more directly causal in a health outcome, such as somebody's exposure to toxins in their environment, right? We should try to measure the things that are most directly causal. We can't always do that. Also, it's exactly because society really uses concepts like race to create social hierarchy that it matters that we understand things like health disparity across racial and ethnic groups, right? And so if we're gonna understand that, we have to use those categories. So, I mean, there are people who definitely argue that we should never use race or ethnicity as categories in biomedical research. I would not fall into that category. I think instead we have to ask, why are we using this category? What are we gonna learn by using this category? Is there a better way to answer our question? And sometimes there is, right? And if we're not using race, like we might also stratify people by SES. And a lot of times when you do that, any association of an outcome with race falls away, and you realize that the association is really with socioeconomic class, howsoever measured. But that's not always true, right? And even if we associate an outcome with SES, we still might need to dig down more deeply to find, real causal connections. So, I mean, I think we shouldn't use race and ethnicity categories cavalierly. We should be much more thoughtful about why we're using them and how we're using them. But sometimes it's completely reasonable to use those categories.
And did you want to answer, Raphael?
Just one quick comment. I completely agree. I think what we try to do to address particularly the socioeconomic element for participation in clinical trials, trials tend to, for gene therapies and advanced therapies, tend to be at large academic institutions. In rare conditions, there are usually only a handful of experts, in a country like the United States. And so as sponsors, we feel that it's really incumbent upon us to minimize any and almost to the extent that we can, any financial component of the participant and or their caregivers' ability to participate in the trial, which would include housing, moving them for whatever period of time is necessary to participate in the trial, certainly in a very intense period. So it's a really important point, and one that we believe that it's incumbent on sponsors to really try and compensate for.
Okay.
Aisha, could I just go back, because you had asked the second question about the geographic distribution of trials that first of all, most of the gene therapy trials, I believe, are in the United States. Then there's a lesser number in Europe and then in China, for instance. But there are very, very few in Latin America anywhere, or South America for sure. Few to none in any country in Africa. And so I think if the question is trying to relate to this question about the use of, say, concepts of race in research, we need to understand that race as it operates in the United States is not the same thing as race as it operates in other countries. So we can't just think that we're studying something about race by going to Africa and then transporting that and saying, oh, so that means something about African Americans, because it might not, right? And so again, I think the question comes down to, first of all, there might be justice questions just around the fact that many people don't have access to these trials. And if these trials provide benefit, then there are large parts of the world where people are not going to get that benefit because they don't get to participate in a trial. On the other hand, I don't think we ought to be thinking about research as primarily having the goal of providing individual health benefit for people. The point of research is to generate new knowledge. And it's a good thing when we can also provide individual health benefit. We can't always do that. And the fact that everybody can't get benefit by being in research is in many cases unfortunate, but not necessarily unjust. I think that we would learn more, and we might develop products that are more generally beneficial if we had trials that were operating in a larger part of the world. And so that is a scientifically driven reason for having better geographic distribution. I mean, I think there are justice reasons to think that we ought to be investing in many different people, right? Whether it's in the United States or whether it's in other countries. And those reasons are not just because we're going to provide direct medical benefit through participation in trials.
Okay, we're about at time. So if you all have like 5 more minutes, we're happy to stay on if that's okay. I do have to do my facilitator plug for tomorrow's event. So we hope that you can join us tomorrow to hear Dr. James Wilson and Dr. Tim Crape speak about the ethical challenges of immunogenicity and toxicity in pediatric gene therapy. If it's okay with the panelists, I'd like to take maybe 1 or 2 more questions because they were so great. So another question that we had was really, how do we reconcile the justice argument that we should devote research resources proportionate to the incidence / burden of disease with the justice issues of rare diseases? So kind of like how they're impacting the population, but also understanding that there are many rare diseases and kind of like the financial benefit and kind of scientific benefits of like the many versus the few. So maybe Raphael can start.
Can I take a quick, I'll take
Yes.
a quick stab just from the perspective. It's the reason I showed the slide from the FDA's website, which is, I think it's best to think about rare conditions in a collective sense rather than, the 30 million number, rather than, for example, the 60 to 100 patient number in the United States with Canavan disease, which is one of our BridgeBio gene therapy programs at Aspa Therapeutics. So I think very much in that regard, it's best to think of rare conditions as a population rather than an individual, than the individual 7,000 plus individual conditions from that regard.
Pilar, do you have any other thoughts on that one?
So I think, first of all, the idea that we focus entirely on the burden as a justification for how much we invest, sort of collective burden, is a very consequentialist argument. And there could be non-consequentialist arguments for why we would show equal care and concern for people's diseases, despite the fact that some diseases, some people who are, as a small group, very burdened by a disease, are still equally burdened to the people who might be a larger group, but are also burdened by a disease, right? But I also think that it's possible that the reasons for investing in rare disease research there are other ethical reasons, not just justice reasons, right? So there are also reasons of beneficence, for instance, for investing in those rare diseases as well.
Okay, and then there was also a question about that gene therapy broadly, but also pediatric gene therapy is really in its infancy. And is it really the time, or is it really appropriate to be focusing on equity when the field by itself is so new? Should we really just be focusing on the technology? And should we just really be focusing on the technology?
I have a very strong view on that.
Go ahead, Pilar, please.
And my very strong view is this is absolutely the time to be focusing on equity, equality, and justice issues, because if we don't, the problems sort of build on themselves, right? Because the more you study a certain group of people and a certain disease, the easier it is to add yet more studies, right? And so if there's a group that is left out early in the game, it tends to be much harder to go back and study them later, or to study their conditions later. So I think this is exactly the time that we should be concerned about equity and equality.
Okay, and Raphael.
I agree. Just to echo that and to say it's part of the reason that I think a lot of us choose to roll things up a bit and to say we, as anyone involved in gene therapy research, whether academic, regulatory, industry, patient advocacy, there's a real advantage to cooperating and being transparent about what everyone's doing, because that collective amount of knowledge, if shared, will, as Pilar said, really rev up the process and get us to scalable treatments for a large number of patients who are burdened. So I completely agree.
Okay. And there was another question around, do you have examples of diversity of rare diseases in marginalized populations? that intersectionality of rare diseases amongst marginalized populations?
Well, there are only, in the US, there are only two gene therapies that are approved. So in that regard, I think not so much. But certainly there's a lot of activity in this space, and so we might be able to in the future. Certainly, it would I would imagine um, we'll find some examples.
So I'm sorry, I didn't mean to interrupt.
No, go right ahead. No, I'm finished.
Repeat that question again.
Do you have any examples of diversity of rare disease in any marginalized populations? So I guess
sickle cell is an example of that, right? I mean, it's not ultra rare, but it's an orphan disease, basically. I mean, there are about 100,000 people in the United States. Well, maybe it's not an orphan disease. But it's, it's a single gene disorder. It's relatively rare. So that might be an example of where it's much more prevalent. I mean, if you're talking about a genetic condition, there's no genetic condition that is sort of completely contained within one group of people, right? Genes spread because people have babies together, right? And so it's never going to be completely contained within one group, but especially very ultra rare conditions. I mean, they might affect, a handful of people. And so that handful of people might be, considered to be part of one ethnic or racial group, right? But I actually don't know enough about the rare conditions to know what other ones might be.
Okay, and you mentioned sickle cell disease, so we'll wrap up with this last question related to sickle cell. So the question is, given the inherent risk associated with phase 1 clinical trials and the history of research abuses in people with African descent in this country, do you think it's a good or bad thing that sickle cell disease is one of the first diseases to be the focus of gene editing clinical trials?
I think it's a mixed blessing, right? In the sense that there is something very positive about, especially the federal government saying, look, here's an area where we have really fallen down in research investment. Here's an area where we haven't demonstrated care and concern for the problems of a particular group of people, and we should do better. So that's all to the good. Whether people experience the risks of phase 1 trials now or later, there are still risks, right? And so, I don't know that that's really a distinguishing feature, of the fact that we're going first. That just is a fact of doing research, that there are risks in research and you have to communicate those honestly, and do some work to make sure that so I had put the phrase governance. Do you have trustworthy governance for the research? And I think that's a huge piece of being able to recruit more diverse populations into research generally, and might be a really important piece of recruiting participants into these, gene editing trials for sickle cell, right? It's like, are you transparent? How are decisions made? Do people know how the decisions are made so that they feel like they can trust you?
Okay. Any other thoughts, Raphael?
Yeah, just speaking brief ones. Just speaking from the respect of from a technical aspect, I agree with Pilar. I think opportunities come up for gene transfer and gene editing based on you heard Pat Furlong say yesterday that dystrophin, for example, one of the largest genes, one of the largest proteins in the body, is a very challenging target for gene therapy. And I think I view sickle cell much more through the lens of it's a more attractive target biochemically, morphologically. And so I agree, it's really incumbent upon the investigators and the sponsors of the research to work with the community and work with experts to understand the dynamics and to address them where there are needs for information. But it's true what Pilar said, the risks of phase 1 trials are pretty standard, particularly in genetic disorders. They're fairly agnostic to the condition, I would say.
Okay. Well, thank you both, and thank you to everyone in our audience. We hope you can join us the rest of the week for the rest of our series. And again, the webinars will be posted on our YouTube channel. So thank you for joining us, and have a great rest of your day. And this is the slide for our event tomorrow. So we hope you can make it. Thanks so much.
Thank you, Aisha.
All right. Bye.
Bye-bye.